we are excited to introduce you to our OmicsLogic training programs in Nigeria. This program will help you see the role of big data and next generation sequencing technologies in life sciences. OmicsLogic Bioinformatics training is designed for everyone interested to learn and apply data science skills, to study biological data sets in an effective and meaningful way. However, before we get started, let me introduce you to our company vision and team. My name is Aditya Aboshade, and I'm the community manager of Omics Logic Nigeria. I'm responsible for daily interaction with thousands of users that are part of our bioinformatics community managed by Pine Biotech. Let me start by introducing Pine Biotech, the company behind Omics Logic. I'll also share with you details about the upcoming Omics Logic programs and explain how the training resources we will discuss today will be used in the upcoming data science training program.
Carry and follow Okay.
So we kindly enjoin all participants to mute themselves so that we will not be disturbed by the noise coming in from their individual phones. The Omics Logic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Toba Bioinformatics Research Center. What we will discuss today and throughout this program is a result of hard work by an amazing team led by the company co-founders, Dr. Alfred Toba and Dr. Leonid Brodsky. Dr. Alfred Toba, who comes from a medical background, has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Toba Bioinformatics Research Center at the University of Ibadan and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career, has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science, and training, and some of them will be presenting today as well. Over the course of the past several years, I've been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review. As a result of this, the Omics Logic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and medical research. The goal of Omics Logic's programs is first to provide training in bioinformatics, enabling independent research guided by mentors and peer examples, developing a growing community with shared passion for data-driven research and appreciation of life sciences, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using the latest technology in life and data sciences. In this webinar, our team will introduce you to resources that will get you started on this journey. We hope that you'll be able to benefit from this webinar. The objective. So let's go to our educational resource platform, which is edutbio.info, and get signed up. Please, Mr. Dekule, can you help me put the website into the chat forum? edu.t-bio.info. Are we all there?
Yes, we have Bipsa Biswas, who is able to put the link in the chat, and we have also Adekunle and Hamza that also are able to do that. So please, someone, please help Adetayo get the link in the chat.
So for the new users, you have to register or sign up. There's an option where I put this black box, which is saying not a member, sign up. If you're a new user, you've not been introduced to the website, this is where you go, you click the register now button, and to bring out these options where you have to input your name, your password. Then you also have to click the I'm not a robot captcha, where you submit, and it takes you directly to the website.
Maybe after.
Is there anybody that have problems with signing up and logging in?
It doesn't look like anyone is having any issues.
Okay then. So in case there are any issues, you can put it in the chat box and we'll attend to you completely. So moving on, we're excited that this event has been led and organized by our dedicated team in Nigeria. It has brought together experts who have an interest in data-driven research and have all recently completed our specialized training. They are now certified and ready to assist all of you in joining this program. Getting registered and finding all of the necessary resources and helping you troubleshoot any technical or conceptual challenges you may come across. Faculty in Nigeria comprises Mr. Adekunle Adeluwoye, myself, and Mr. Sonny Hamza. And this faculty will be supported by our team in the US and India. We have Dr. Apit Kaur, a certified Omics Logic trainer and expert bioinformatician, who will guide the hands-on sessions and assist the participant with projects and analysis during the program. Our area of specialization is cancer genomics and machine learning. We also have Dr. Mohit Mazonda, Omics Logic project mentor and an expert in big data computational analysis. Dr. Mazonda has a PhD in computational biology and over 10 years of experience working with industry and academia. We also have Elia Brodsky, Biotech co-founder and CEO. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. I will now pass it on to Elia to talk about the increasing significance of data science in biomedical research and introduce you all to the types of challenges we will be addressing in this upcoming program. Over to you, Elia.
Thank you very much, and welcome everyone to this webinar. So as Adetayo mentioned, I am one of the co-founders and currently the CEO of Pine Biotech. And as a result of being in this role, I have gotten a lot of experience interacting with participants of different training programs, as well as researchers from around the world that are using a lot of our infrastructure to help them manage and analyze big data. So this webinar is the second webinar. The first one that we held a few days ago was dedicated to the very first introduction of the different resources and the topics that we will cover in this upcoming basic Omics Logic training program for Nigerian participants. What we asked all of you to do last time is also to get signed up to make sure that you understand how the materials are organized. And so we were excited to see that many of you have also completed your profile, started learning by taking some of the coursework that we asked you to complete, and you probably noticed that you have points for completing different types of courses and activities. You also have an indicator on your profile page of how much of your program, of your course you have completed, and also you have a certificate that comes with many of the courses that we have available. So that's what we did last time. We also asked many of you about your educational qualifications. We saw that the majority are master degree students, and the second bigger category is bachelor's or undergraduate students. We also asked about research interests, and we noticed that many of you were interested in basics of bioinformatics, infectious diseases, and precision medicine. And we also asked about what type of omics technology you're interested in, and it looks like the majority are interested in genomics. And typically that happens because many people are much more familiar with the topic of genomics and with the term genomics rather than some of the additional omics data types like transcriptomics and metagenomics. So this is some indication for us that helped us shape today's presentation and talk about the different technologies and how do they all combine together to illustrate how we can study different types of biological phenomena using bioinformatics. So what is covered in this Introduction to Bioinformatics course? The Introduction to Bioinformatics is just one of the introductory courses that we have, and it introduces participants to different applications of bioinformatics and what bioinformatics is. But we also have these introductory courses for different types of omics data. And we've seen in our previous programs that many of the participants are highly rating these resources that are intended for asynchronous independent learning. Let's talk about what bioinformatics is and how is it related to what we will cover in this overall training program. Bioinformatics. Aditayo, if you could please help me by, yes, keeping it quiet. Thank you. Bioinformatics is the intersection of informatics, statistics, and biology. It's like a specialization of information technology and statistical analysis for biological data and biological problems. And this includes a variety of different skill sets that we try to cover in our introductory program to get everyone on the same page and provide a perspective for how to address different types of biological challenges. In the world of biological data, a lot of things have been happening over the years. So you can see in this next chart how the amount of data and the variety of data is really changing at a dramatic pace. As a result, many biologists and clinicians are facing this problem of big data and how this data comes in almost real time, as we have seen in this pandemic. Almost every day, there's new information based on data about genomic variation in the viral genome. We see different types of host responses, patient responses, and different animal model responses to how different treatments are applied or different infections happen in almost real time. So we are really seeing an influx of a variety of different omics data types. What kinds of omic data types are there? There's phenomics, or characterization of cell morphology and tissue specificity, and essentially what are the external characteristics of a sample. Genomics, which many of you are probably familiar with, or what is the genomic variation or DNA code variation that we can study at the single nucleotide level or ranges of nucleotides or regions of the DNA. Epigenomic variation, how the condensed chromatin inside the nucleus could be expanded, become more available and ready for transcription, and how non-coding RNAs are guiding the process of gene expression. And finally, the gene expression itself. How can we understand the expression of different transcripts in the context of different conditions and different samples that we observe? So the unique part of all of these omics data sets is that they have to be processed. So essentially, the data comes in a form that we need to take significant steps to modify from short reads into a table that could be analyzed. And this is true for genomics, epigenomics, transcriptomics, and proteomics, or metabolomics, so many different types of omics. Ultimately, however, this whole field of omics data and bioinformatics is becoming more and more focused on the challenges of integration. So multiple factors that cause disease and lead to disease progression lead us to the omics integration process. So we need to look at how cellular factors, microbiome, the virome, and clinical and phenotypic data are all speaking about the same phenomena. And so as a result, we have designed these Omics Logic programs that are focused on individual types of omics data and individual types of clinical and phenotypic data in the context of applications. How can they be applied to infectious diseases? How can they be applied to oncology? How can they be applied to microbiome, etc.? So how are these programs organized? The way we conduct these programs is by leveraging online tools like Zoom, as we're doing today, where we present a specific topic and have a discussion around the topics related to this problem. Self-guided study materials that help you understand a topic in greater detail and learn about the different applications from a theoretical point of view, as well as some practice assignments and interactive practice of what has been learned. Okay. You could please be mindful of people that are joining so that when they join, they could be muted. Thank you. So when we have these practical assignments, and by the way, we'll do a couple of these today. So those of you that have already registered on the Introduction to Bioinformatics course, you have probably been introduced to the T-Bioinfo platform here on top. And we'll look today at a couple of pipelines from the T-Bioinfo platform, and also the practical coding assignments that come on our Omics Logic Code Playground. So I'll introduce those in detail in a few more slides. So how can we apply these different computational tools to data sets based on a specific topic? What we do in this program is we help link the different types of conditions that are described in projects and publicly available data sets to the type of data and the methods that come with that data. For example, we'll talk today about 3 different types of resources that we will leverage in the course. The courses on EDU T-Bioinfo portal, the platform, and the coding challenge on the coding platform. So let's take a look at one example. One obvious example that most of you were most interested in is genomics, right? So how can we take DNA variation that describes a condition like cancer, so these have to be somatic mutations, and process that data so that it could be used for analysis and understanding of that biological condition. So what I'll do now is I'll go over to the browser and I will look at the browser for what we have in one of our courses called Genomics 1. And in this course, Genomics 1, there's an introduction to somatic mutations. Okay, so if all of you are already logged in and registered on this portal, you will see here under courses, Genomics 1. Okay, so please let me know when you are there by putting number 1 in the chat box so that I know that most of you have found this resource called Genomics 1. Okay, so I see that Deborah, Adetayo. Okay, anyone else has found this resource? And maybe we should post here the link. Okay. So this is the link to this resource. Please let me know once you have found it and it has loaded. Let's take a look at what is covered in this course. So it starts with what is genomics and leads on to talk about the biology of the genome and define what is genomic variation. And in lecture 2.2, you're introduced to a pipeline, and this pipeline allows you to link together multiple scripts that will transform raw data from a sequencer into some kind of a defined genomic variation study. So let's take a look at what that will be like by going to server.t-bio.info. Okay, so now we're looking at two things at the same time. Okay, we're looking at the course that introduces you to genomics. And from that course, you are introduced to a bioinformatics analysis pipeline, which is on server.t-bio.info. Okay, so how many of you have now been able to see this pipeline? Please put one in the chat box so I know that you've gotten there. Okay, and Beepse, if you could please post the demo information on how people that don't have an account can join and see the demo pipelines. Okay, so now we have the username and the password. If you want to try this out and see how it works, you can go right here, log in. And once you're in, you'll be able to view some demo pipelines that will illustrate how to use this pipeline. Okay, so what I'll do is I'll show you a couple of examples today. So you can see that this whole section belongs to next generation sequencing data. Next generation sequencing data means that it was data generated by a sequencer that produces short reads. And those short reads could now be analyzed for variation by mapping them onto the reference genome. So let's see how this works. Right here, if you scroll down, we will get to genomics epigenetics. And here we have a section called mutation variant, parallel analysis of mutation variant data. This is the pipeline that is used in Genomics 1. So here we can click on start. You will see that we are analyzing variation at the single nucleotide level. And we can pre-process and map this data. We're not going to pre-process for now. We're just going to go to bowtie 2, which is a mapping technique. And after mapping, we will go to visualization. So visualization will allow us to see what do those short reads look like when they are mapped onto the reference genome. Okay, so after I have completed this pipeline, I've prepared this simple pipeline of several steps, I will be able to click on visualization. And visualization will show me how those individual reads are mapped onto my reference sequence or the reference genome. So you will notice that I have here two samples, and these samples are going to be displayed over here, the reads from those samples. And you can see that it says too much data to show. Indeed, this is a big data analysis. So there's a lot of reads in each one of these. And as I zoom in, I am starting to see how the individual reads are mapped to the reference genome. So as I look in and I keep on zooming in, eventually I get to a point that I can see the individual nucleotides on the reference genome and the reads that show me the coverage of what I have coming from these two samples, right? So here I have group 1 and then I have group 2. Now, these are two samples that represent a cancer sample. Essentially, if you think of a patient that has a tumor, for that individual patient, what we need to know is that patient's tumor genome and that patient's general healthy genome. And by comparing the two, we can separate the germline mutations from the somatic mutations. And that's exactly what you will see as you do this pipeline and this analysis. So the platform really allows you to explore huge data sets. Again, this is whole exome sequencing of a cancer patient. And by comparing those genomes, we can find somatic variation or specific genomes that are responsible, genomic variations that might be responsible for tumor differences from the healthy genome. Okay, so I see that there's a raised hand, but I would ask that you, instead of raising the hand, if you have a question, please type it into the chat, and we will be looking at those questions in the chat in just a few minutes so that we can help answer any questions that you might have. Okay, so if you have any questions about this pipeline or about genomic data, please go ahead and put those questions in the chat box, and we will go through those questions in just a second. So what did we see in this example? What we saw in this example is how we can take a genomic data set from whole exome sequencing and process it through this pipeline to get an understanding, a visual representation of read alignment to the reference genome. Now, how many of you know how many nucleotides there are in the human DNA? How many people remember how many nucleotides are there in the human genome? All right, thank you. Gio Oboli is saying 3 billion base pairs. Anybody else? Okay, 3.3 million or billion, 30,000 billion. Okay, so there are some maybe different opinions about how long it is. So most of you are correct. It's about 3.2 billion is the actual number. So 3.2 billion. So it's a huge, huge sequence, right? And what we are doing here is we are aligning short reads that come from the sequencer to the reference genome. So how long do you think our sequencing read is when it comes from the sequencer? Does anyone know the length of a typical sequencing read? Okay, for those of you that are just joining, we're discussing genomic data and how sequencing can help us study genomic variation. And we just went through an example of a pipeline that helps us process data that comes out from the sequencer in terms of genomic variation from the reference genome. So the question is, how long is a typical read? Okay, 100 base pairs. Somebody is saying maybe 60, I don't know, seconds, but maybe 60 base pairs. Okay, so the answer is it could be anywhere between 30 and 350 base pairs. Okay, that is the next generation or the second generation sequencing technology, between 30 and 350 base pairs long. So as you can imagine, using that kind of sequencing technology, you end up with millions and millions of reads that are aligned to a super long sequence of the whole reference genome. And so computationally, this is a very intense process that requires what's called high throughput computational analysis technology. So it's a parallelized analysis of many of these sequences and follows a complicated process that will allow us to understand where do those reads fall onto the reference genome and how to find significant mutations. Okay, so here we have a series of courses, Introduction to Genomics, Genomics 1 and Genomics 2, that cover a lot of these topics. They cover the topic of how is this data prepared, how does the mapping happen, how do you prepare your own pipeline, and how to make sense of what you get out of this pipeline. Another type of topics that we discussed in our last session, and many of you showed an interest in, was transcriptomic data analysis or gene expression. How can we use the same technology to understand variation at the gene expression level? So here you can see another pipeline that looks somewhat similar. Again, it's a series of scripts that are connected together. And what they produce is what's called a table of expression. The table of expression is a process where we take individual reads and count how many reads we have for each one of our genes to understand variability between samples, as you can see in this principal component analysis, that maybe is described through the gene expression levels. Okay, so here you will go through another series of courses. Okay, and let's take a look at these courses. Let's take a look at what we have for gene expression data. So again, please go with me to the EDU T-Bioinfo portal. Okay, and on this portal, if you go to courses, you can go to Transcriptomics 1. This is another course, just like we look right now at Genomics 1. Okay, we can look at Transcriptomics 1, an introductory course for transcriptomics. Thank you, Bibsa. So in this course, you will find a whole set of introductory materials about gene expression. What is transcription? How to quantify and understand portions of DNA that are being expressed. How can we then generate next generation sequencing data? And also will provide you to a pipeline. So here you will see how to develop an RNA-seq study. Okay, so the RNA-seq analysis pipeline, we can look at it again as a demo. And Bipse, if you could again post the demo details in the chat box so people could look at those. Okay, those that are joining just now, we're providing you with just the demo details so that you can try to analyze the data as we go through this example. Thank you, Bipsa. Okay, so here again, we have a section on next generation sequencing data. The next generation sequencing data can be used to study genomics, like we just looked over here, or can be used to study transcriptomics or RNA-seq, how we can measure and quantify gene expression levels. Again, to look at this from a demo perspective, we can take the same data and build a pipeline that allows us to study variation at the gene expression level. So again, we can click on Start. We will do some pre-processing by trimming our reads from all kinds of technical sequences, analyzing variation as a result of PCR bridge amplification and removing those. And then we can map our gene expression data or our sequences of reads. We can click on bow tie to T and prepare an RSEM table of expression. Okay, and this is what the table of expression will look like. So once we put this together, this is again a pipeline. This pipeline will show the following results. These results will include the table of expression, and that table of expression could be in different formats. It could have expression of genes, expression of isoforms, expression of exons. So once you see this type of data, you will realize that it has a lot of information. How many of you remember how many genes there are in the human genome? Okay, so if you know the answer, you can put it in the chat box. How many genes are there in the human genome approximately? Okay, does anyone remember how many genes? Okay, thank you, Avoli. About 20,000. Tofik is saying about 5,000. Okay, anyone else? Adetayo is saying 30,000. Okay, so indeed, the right answer is about 25,000 genes. Okay, so 25,000 genes in your typical human genome. Now that number is approximate. Why? Because the definition of a gene is a sequence that produces a mature mRNA that will be translated to protein. So when we talk about genes, that definition and finding those sequences has been changing over the period of time. As you know, the human genome was fully sequenced about 50 years ago, but for the past 50 years, that information has been going on, and we know now a lot more about genes and about their function than we did back 50 years ago. So this is a dynamic field where a lot of changes are happening. So if you open this table, this is a table of samples used to produce the table of expression. You can see that this is breast cancer genes or breast cancer samples of different subtypes. And so when we will expect our table of expression, we will actually have 58 samples, and those 58 samples are going to be of different types of cell lines. So the process of now downstream analysis of the data that we produced in this pipeline is going to be again explained in our online coursework. Okay, and so let's take a look at what type of coursework do we have for transcriptomic data so that all of you can gain insight into what to do with this table of expression. Transcriptomics 1 will discuss the process of analyzing the individual reads and transforming them into a table of expression and how to explore the table of expression. Transcriptomics 2 will talk about statistical analysis of differences between groups of samples. Transcriptomics 3 will introduce you to machine learning, and Transcriptomics 4 will show you how to use this for single cell transcriptomic data. Okay, again, if you have any questions about transcriptomic data analysis, you can put that in the chat box and we'll be coming to your questions in just a few minutes. Another type of data that a lot of you have shown interest in is metagenomics. Metagenomics can help us study the microbiome, or the complex community of microbes that lives together in a single sample, to understand differences between sample groups based on species diversity and richness. And this is an amazing change in the way microbes can be studied. First of all, microbes were first detected as a result of looking through the microscope. And as the resolution of the microscope has increased, we now know that these are complex communities. You can see here an example from a very high resolution microscopic image that shows how individual E. coli bacteria are being attacked by phages. And you can see the individual phages right here. So what people have now understood is that this is not just a single organism that lives in isolation, but this is an interacting community of different microbes that live together and work together to produce what we call a phenotype, whether it's a condition or a disease. And so from the isolation techniques in the lab, today we can look at a single bacteria, or we can start studying these bacteria as a microbiome, as communities that function and live together to produce some kind of an environment that could be associated with a condition. And so when we talk about the microbiome, there are a growing number of papers that are being published on the microbiome, including the lung, vagina, brain, skin, liver, and other types of mouth and intestinal microbiomes. There are literally tens of thousands of publications on the way microbiome could characterize different diseases. And so we can look at these ongoing projects and studies to understand how to analyze such data from publicly available repositories to study disease of the gut, sensitivity, different types of skin conditions, lung conditions, even brain conditions. So a lot of different data sets are available out there. So again, if you have any questions about the microbiome, you're welcome to post those questions in the chat so that by the end of this presentation, we'll be addressing many of your questions. Another important part of this training that we're going to be doing in the upcoming program is how to analyze these large data sets once they are processed and structured. So we will look at a number of different examples of genomic data, transcriptomic data, metagenomic data. Each one will come in a structured format. It will be a table. And we'll look at different applications of machine learning and statistical analysis methods to understand how we can extract meaningful insights. And for that purpose, it's important to have some introduction to coding skills in R and Python. And so maybe somebody at Adedayo or Bipsa, you guys can post the link to the coding platform. On the coding platform, we have practical exercises for those of you that are ready to analyze such data using R packages and Python approaches, where you can learn about things like principal component analysis, different visualization techniques, classification, clustering, etc. So you can really take a look at that. And again, you can sign up for free. As we will go through this program, we will leverage some of those exercises in our upcoming sessions. So all that we covered today is a good overview of what we will in detail study throughout the whole program. That will include a number of different sessions that will focus on one particular topic and give you a lot more time to go through understanding from very beginning up until you know how to apply this information to a particular data set. So going through this training will give you the tools and the conceptual understanding to start using omics data and bioinformatics. So this will include a session on introduction to next generation sequencing. We'll talk about the different file formats and the links and the different specific details about this data format. We'll talk about bioinformatics in application to big data and biomedical research. We'll also talk about how we can leverage the asynchronous courses in greater detail. But it's not just about the asynchronous courses, you also gain access to mentors that can support and answer specific questions you have. And then we'll go through individual types of data and do a little bit of practice together to understand how to use those data sets effectively. Now, as we go through this program, you will see that your activity and everybody's activity that is participating in this program is going to be tracked. And the reason for that is we want to find a most effective way to combine the practical exercises on the platform and on the coding playground with the asynchronous theoretical courses so that everyone can start working on independent projects. So these independent projects will allow you to apply what you have learned to a data set that has a description of whatever that project was. And you will see how after you have completed the basic training, you will gain access to practical examples that are sourced from peer-reviewed journal data sets that have been published in association with a real challenge. So we'll look at such examples as Ebola virus outbreak in West Africa and the origins and pathogenesis of the novel coronavirus. So case examples, case studies, and examples that are very relevant and current with what we are experiencing as citizens and also as people interested in biomedical research. Now, we have done this program with thousands of students. We encourage all of you to sign up, but just to give you a perspective on how this program has benefited other people in Nigeria that are going to be helping you go through this program, I wanted to introduce you to our OmicsLogic Nigeria team, which includes Adekunle Adeluwoye. He's an OmicsLogic Nigeria project lead, and he's a faculty at Leeds City University. Adetayo Aborisade, he's also faculty at Bayero University in Nigeria. And Sani Hamza, he is a medical laboratory scientist at the University of Lagos, Nigeria. So Adekunle, I would like to pass it on to you now. Please, can you hear me? Okay, as Adekunle is joining us, I want to just briefly take a look at the questions that you posted in the chat box. I see that some people were asking for the video recording, so I'm sure that Bipsa is going to share that on our YouTube channel. And then I see I see. Okay.
It's me though.
One second. Okay.
So earlier, there is this question by Farida Bashir. Can you hear me?
Yes, I can.
Oh, our question is, can a PCR machine serve a function as the next sequencer while you want to perform gene analysis of a particular disease?
Well, PCR machine is a process of generating sequences of a particular kind of determined type, right? So for example, you can have a primer, and that primer amplifies RNA or DNA in your sample. So that way you can study specific types of sequences to identify whether they are present, and if they are present, if they have some variation. So it's a good way to find mutations. It's a good way to analyze specific types of sequences. Next generation sequencing is different from PCR itself. It does not just amplify the genetic material, it actually allows us to study all of the genetic material in a sample and quantify it, including looking at specific variants like mutation variants. So I'm not sure that I have answered that completely, but I will take a look again at all of these questions towards the end of the session. And in the meantime, Adekunle, can you hear us?
Yes, please. I can hear you.
Okay, great. So I'll pass it on to you then.
All right. Thank you very much, Elia. All right. As mentioned, my name is Adekunle Adeluoye. I'm a faculty from Leeds City University, Nigeria, and I'm also a mentor with Biotech. I have my background in medical laboratory sciences, and my area of focus currently is molecular biology and genomics, where I'm currently completing a PhD degree. So as a lecturer, I'm involved with lecturing body medical scientists, nurses, and radiographers, all of which are taking courses in the biological sciences. And I can say authoritatively that many of our curriculum is designed around the theoretical component of this course of studies and has less to do about the analysis and utilization of biological data that is being generated in our clinical and laboratory settings. As many of us know already, the volume of this data set has increased significantly in this era of genomics and omics science. And like I did mention last time, as exciting as bioinformatics is, the fact is there are computational resources gap when it comes to successful running of these bioinformatics pipeline. This, like we said, includes infrastructural requirements, of course, the cost, and we also have inaccessibility issues. But more importantly, which is one area I'd like to focus on this evening, is the expertise gaps. Our teachings in its present framework within our universities does not teach required data science or bioinformatics skills, which are skill sets that is needed by our students to adequately analyze this data and to make insightful discovery from them. And this is where personally I find very interesting and helpful the Biotech's cloud-based artificial intelligence guided server for participants to utilize in their learning journey. Aside the well-structured asynchronous courses which we've been introduced to by earlier, and other learning resources that are loaded on the website, the learners can immediately begin to have hands-on experience on big data analysis. And so by that, I mean, as you are learning, you are having opportunity to practice what you are learning using, the available resources, especially the T-Bar server, that is being provided by Biotech. Here on this slide is a screenshot from one of my own personal project pipelines, which I run using the cloud-based AI-guided T-Server Bio platform. And it will interest, many of our participants to know that I, got involved. I mean, I came across Biotech sometimes last year, and you can imagine what I've been able to learn using the platform within this short span of time. Okay, so I want to emphatically say Biotech platform, the EduTPAL info platform, as well as the TPAL server platform are two platforms that will help each and every one of us to learn bioinformatics, big data science, and as well as apply these learnings as we go, on in our learning journey. So I therefore look forward to having many, more professionals, university faculties and lecturers, researchers, as well as students as budding professionals, in the making to come up and key into the opportunity that is available by participating, in the program that is forthcoming, the basic Omics Logic training part 1. Of course, as facilitator earlier spoke about, there are a series of programs that are to come after the basic Omics Logic training part 1. Okay, but sequentially and methodically, we want to use these programs to introduce participants to these skill sets in the bioinformatics and big data sciences, and to ensure that participants get to learn these skills and able to utilize it in their own personal researches. Quickly, before I hand over to the next speaker, I'd like to mention from our last session, some of the questions we have, that people were asking has to do with asking the possibility of applying what we are here learning or talking about in their own personal, studies, in their own personal field. We have people talking, that they are in the ecotourism and wildlife, field. Some are from animal technology, while some are, equally clinicians and pharmaceutical microbiologists. So I like to say that, learning these skills, apparently, we go a long way in enhancing our researches and the way, we utilize the data sets that are being generated in a laboratory. And quickly, another thing I'd like to add, which I know came up in the last webinar we had, was trying, some people asking about possibility of making this training, the main training, which will be starting next week, Tuesday, that is on the 19th of January, making it, cheaper, as it were. And well, thankfully, earlier mentioned last week that there's an opportunity for scholarship for some of us who might be having challenges to, make the payments for this training. Howbeit, I like to mention that these same trainings in other climes in Europe, in America, costs, about $150. Okay, and we've been able to, discuss and prevail on the leadership of Biotech to make these course, offer it to Nigerians at $25, which is equivalent of about 10,000 naira. But of course, I would like to assure all the participants who will be coming on board that we're going to be getting much more than the value we are putting in. The last thing I'd like to talk about before I hand over will be the access to the resource materials. Yes, we have a number of free resources on this platform. Introduction to bioinformatics, which we expect all our registered participants, as well as just everyone on this webinar to register a profile and try to run through these courses. But outside of that, people who go on to register for this training program that is forthcoming, we have access to the server that we've been talking about, the server platform, with which they will have the hands-on experience, and of course the mentor and the support that will be needed to learn and practice what is being learning on the go. So on that note, I will want to hand over, but encourage as many of the participants who still have one or two questions to please not hesitate to ask their questions so that we can answer each and every one. At this point, let me pass it over to Dr. Aborishade to take it up from there.
Thank you, Mr. Adekule and Ilya. I think for me as a clinician and as someone that is interested in research, especially in our climes where resources are limited, the first question that I asked myself when I wanted to come into bioinformatics is the cost. Can I afford sequencing? Can I afford next generation sequencing? Can I afford to do all these research items by myself from the scratch? And I think that's one of the questions Ilya already answered. As researchers, we really do not need to start most of these studies ab initio. There are studies that have been done, sequences that have been placed in repositories, public databases, that as a researcher, you just need to know how to retrieve this information from these databases and just start your analysis from that start. That's also one of the advantages of this training program. That's one of the advantages of this platform, that you can be guided by the facilitators and the mentors on how to retrieve information from public databases. For example, it could be genomic sequences, it could be transcriptome sequences, how you can retrieve them in your field, in your area. If you're a veterinarian, you can receive genomic sequences or transcriptome sequences of your research animals, like dogs, like cats. And the only thing that you do as a researcher is just to analyze them and be able to exact the same questions you would have asked if you are sequencing them ab initio. That's also one of the advantages of this training program. Another advantage is, as novel bioinformaticians, there is a dearth of mentorship, especially here, due to the expertise gaps. One of the problems I had when I started was I needed mentors to guide me as regards project tools and research items, which I did not have at those times. It discouraged me for a while, and it nearly killed the love of bioinformatics. But that's one of the advantages Biotech also has, that both locally and internationally, you can speak with the faculty in Nigeria, you can also speak with the faculty in US and India, and each and every one of them will be eager, will be available, and they are approachable to assist with whatever research item or research tool you needed at that point in time. Another thing which I also found that made it easier to learn with bioinformatics is that you do not need to be a connoisseur in data science. You do not need to be an experienced bioinformatician or a machine learning person before you can make use of the pipeline. If you can follow the steps written out in the asynchronous courses and the coding platforms, you can as well code, you can as well practice like a veteran bioinformatician, because they are well spread out. They are written out in A, B, and C's, in 1, 2, and 3, giving you the desired outcome for each data or research tool you have. These and many more are the advantages of these programs. And the reason we also feel that, aside the faculty in the US and in India, that we also require a faculty in Nigeria, because we can, so that at every point in time, you can easily put a call through to any mentor. You can easily put a call through to any faculty in case you need queries, questions, and guidance and mentorship as regards any of these pipelines. And that's why we feel that this program is well equipped to answer and cater for every of your research item, every of your research questions, every of your curiosity as regards bioinformatics, so that by the end of this training program, you can have a full established faculty in bioinformatics and data science. Thank you very much. I'll hand it over to Ilya at this point.
Okay, thank you very much, Adekunle and Adetayo. And again, the team in Nigeria is going to be supporting a lot of your questions and technical issues, but I want to answer some of the questions that have been posted. Taiwo has asked about his research problem. How can this training aid and promote my research? And other people have specific questions about their projects and their data sets. And so, as you have seen, we've taken a few examples of published data that is on NCBI translated those into project examples. And a lot of our participants in previous programs have been able to use that as a springboard into the world of omics data and bioinformatics. And I want to give you just a couple more examples. So here is Dr. Ayurinde Fayehun, who's also a physician and is doing his master's in bioinformatics at University of Ibadan in Nigeria. And after completing the training that you can see right here, the certificates that he received from the training program, he developed his own interest in infectious diseases, found a data set on NCBI under the guidance of our team, and was able to perform a differential gene expression analysis to identify genes responsible for response to infection or the immune response to infection. Another participant of one of our previous programs, Kuda Kwashe Nyombo, who is a researcher and has a Master's of Science and Technology from University of Zimbabwe in Harare, was looking at in-depth transcriptomic analysis to determine the response to inhibition of PKNA gene and PKNB gene by tuberculosis. So as you can see, for any of you interested in a particular domain, the limiting factor for you to be able to apply bioinformatics to a research project of your own is not the tools, and it's not the understanding of the technology, it's actually just availability of data. So before we continue to speak about all of the rest of this presentation, I wanted to take you to a source of data that you can find on NCBI. So please come to this website. This is the National Center for Biotechnology Information, a website dedicated to different types of data. And after you get to this website, please let me know by putting in #1 into the chat box. Okay, so please go to ncbi.nlm.nih.gov. Again, you should see something very similar to what I have right here on my screen. And thank you, Deborah, for putting in #1. So that means that you are there. So briefly about NCBI. The National Center for Biotechnology Information is a collection of different types of resources. You can see different resources right here. They include data and software, different types of software, and different types of structures and data sets. Okay. All right, so I see Honor is also here. Okay, so for those of you that are here, you can see that we can search in here for any topic that you would like. So would anyone like to propose a topic that you're interested in? What kind of an experiment would you be interested to look at to understand if they have data about that experiment? Okay, so you can just put that example into the chat box again, for those of you that are already here. Okay, any topic that comes to mind, any topic that you're currently researching, we'll just want to see whether they have this type of data on NCBI already. Okay, hepatitis. And Hamza was saying hepatitis. Farida is saying serine protease of snake venom. Okay, anyone else? Let's do just a third one. So if anyone else has an idea. Okay, diabetes, sickle cell anemia, antimicrobial peptides, cancer genomics. Okay, so let's take a look at these examples one by one. So the first one was hepatitis. Okay, and I do want to mention that the program is designed in such a way to introduce you to the different types of omics datasets, introduce you to different project examples that we have prepared, and allow you to work on an independent project of your own. For those that complete all of this training, we will provide a certificate of excellence and a platform for you to speak about your project to an audience that is probably beyond your reach right now, an international audience that will listen to your project example. And so these milestones that you will achieve throughout the program, the basic training, the introductory materials for projects, and a project of your own, each come with a specific certificate, overall turning it into a certificate of excellence. And a certificate of completion of these courses will be given to each one that completes the basic training, okay? So here we see the results for hepatitis. Now, obviously, there are many different types of hepatitis C and B in different animals, etc. But you can see this is gene expression omnibus datasets. So how many datasets exist using transcriptomic data? Over 9,000. You can see right here raw data, sequence read archive, so just reads from the sequencer, over 12,000. Bioprojects, over 700 bioprojects. So these are collections of data sets, okay? So a huge amount of data. Let's take a look at another one. Someone mentioned snake venom, okay? So let's put this snake venom. Again, geodata sets, over 20 geodata sets. Over 30 SRA read archives. Let's just take a look at some of these examples. What can we find here? You can customize your search, and we'll go through this in much greater detail when we do our program, but just quickly for the purpose of today, I'm going to change this to expression profiling by high throughput sequencing. Okay, and now I can click on this filter. And let's see, so here's a gene expression data set. The neuromuscular junction is a focal point of mTORC1 signaling in sarcopenia. Okay, and this was, you can read here a little bit more about this abstract. Okay, so not sure that this is directly related. Let's see over here. RNA sequencing of venom glands and venom gland organoids. Okay, I think very interesting. What else do we have here? Okay, so you can see that we have quite a number of results for snake venom. Sickle cell anemia, so such common conditions like sickle cell anemia, hepatitis, there's probably going to be a lot. Okay, here you can see 579 items for sickle cell anemia. So single cell transcriptomic profiling provides insight into maturational and developmental status of pluripotent stem cell derived erythroblasts. Let's see, transcriptional responses in children with sickle cell. So a lot of different examples exist for a variety of topics. And I think that, you know, importantly, we are all now captured at home, sitting, waiting for us to understand what can we personally do in this era of this pandemic, of the rise in infectious diseases. How can we apply bioinformatics and publicly available tools, as well as data sets, to make a difference, to make an impact on what's going on right now? And I think that exciting part of what we are doing here is not just learning for the purpose of gaining certificates, right? We want those that complete this training to be citizen scientists, people that can contribute solutions and new ways of analyzing data to understand what's going on around us. So let's take a look at an example from another resource from NCBI called NCBI Virus. Okay, so NCBI Virus is a repository of data sets specifically about infectious diseases, okay? So in this repository, and I'll just put the link over here, you can find a whole collection of resources dedicated to communicable or infectious diseases. Okay, so for example, let's search by virus, and we'll put in here SARS-CoV-2. Okay, so you can see I put in SARS, and it's going to open up a drop-down. And you can see the first one is severe acute respiratory syndrome coronavirus 2. So I can click on this to see all of the associated resources. And you can see that there are over 48,000 nucleotide sequences available to study related to SARS-CoV-2. They are organized here by type of virus. You can see the sequence length and type. You can see nucleotide completeness, and you can also see in which coast and which geographic region, including over 400 in Africa. Okay, so over 400 sequences for SARS-CoV-2 in Africa. So I think in this program, you will realize how learning about the tools and about these kinds of repositories really opens the door for you to start thinking about an independent project that could be very relevant if you're involved in any kind of biomedical research to make a difference and not only learn about this for your own good. So again, we have a lot of examples of people that have successfully completed, also have gone on to receive certificates of completion for the full program and develop, for example, you can see here the research project developed by Kingstone Nehaka is SARS-CoV-2 evolution, spike gene mutations across geographical regions in Africa. So I think I will say that's exactly, sorry, the type of question that you were asking. Okay, so research projects, right? I think what's important here is that you will gain access to training and tools, and then introduction to a number of different projects that you can try to apply these tools in a guided way. So for those of you that have already signed up on the platform, you need to now just join the course that we are discussing in this webinar. For those of you that have not signed up, what you need to do is you need to register now. Okay, so let me show you again how to do that. Let's go back to this website. Okay, so please let me know. For those of you that have not registered, you need to be able to see on the main website a login button. Okay, so right here, if I am already registered, I will see my name. If I am not registered, I will see a login button. So if you have already created an account, and please complete your individual profile with your full name, because that will go in your certificate, and an avatar or your picture, because that will help us understand who we are dealing with and others to see that you're a real person. Once you get there, right here under events in the main menu, you will find different programs that are available. And the one we are discussing in this webinar is this one for Nigeria basic training. So you can click on this button and you will be redirected to the organization page for those that will participate in this program. Okay, so what does this organization page include? It includes a forum where you can post any of your questions or any of your challenges, technical challenges, courses that are included in this program that will include genomics, transcriptomics, and metagenomics, projects like Ebola virus and SARS-CoV-2 and others that you can start exploring, a full syllabus of the program, assignments that you will be graded on and will help you understand how much you are able to do with the types of tools that we are doing, and a few other things. So all of your activity will end up being listed over here, and you will be able to interact with the other participants. And we have hundreds and thousands of users that have completed this training, and we want to organize a community out of this training, because it's important that when you're in bioinformatics, you can interact with others that can think of new problems, new challenges, new data sets, and applications to real problems, right? So what we have is a community on Facebook. We have a Facebook group called Learning Biomedical Informatics. And we also have a LinkedIn group. So you will see that joining this website is an introduction to a community, not just the training. And you will see that in these communities, we also highlight the profiles of those that do exceptionally well in the programs, develop impactful projects, And that helps you form collaborations, maybe even get admitted to a lab, a research lab, or find different opportunities for employment and building out your career. So we hope that you can leverage this as a part of this training as well. What we will help you do is understand the specific topics, develop a project, and also present this project in an impactful way to your peers for peer review in the program, potentially also going on to publish in a separate program. Those of you that are just interested and you are still considering signing up, we have a number of opportunities to join different types of programs. And for those of you that already have the training, you're already a bioinformatician, you're already good at doing the analysis part. If you would like to work on a project of your own, we have a special research fellowship, a program that is designed to help you develop a project. And this could be a 3-month or a 6-month program. In that program, you will work with a mentor to go through the important parts of literature review, planning your analysis, and preparing your presentation so that you have an impactful project at the end. Again, we welcome you to join the program. This program is going to start, Bipsa, maybe you could share some of the practical details on how to sign up and when everything will get started.
Right, Ilya. If I may share my screen, I do have certain things to show everyone.
Okay.
Thank you, Ilya. Yeah. So as you all know that we are starting the program on January 19th. So this is the landing page. I'm sure that most of you must have registered as you're joining today's webinar, but in case you have not, I've put the link in the chat box and I will put it down again. So this is the first step to get started by registering. And once you do, you will receive an email from africa@pine.bio. In case you want to reach out to africa@pine.bio, you can also email the team right there. And once you're registered for the program, you have access to this organization page that Ilya has been showing you. Now, I did explain this in our last meeting. So today I wanted to talk about what should you do once you are registered for the program, you have made your payment, and you have received all the info. The first step that you should do is update your profile. Now I'm going to be showing you 2 examples of different profiles. Now here I'm taking Kahinde's profile as an example. So I see that Kahinde has full name written down, completed Metagenomics 2 and has earned 600 points. But however, the profile is incomplete. And on the other hand, Ronald here has a complete profile where you can clearly see that this is a person, and you can identify Ronald from this profile and see that completed all these courses. And the reason completing your profile is very important is because as a community, we can then reach out to each other, because this is a scientific community and we believe in collaboration. And I'm sure that when you're part of this program, you will be collaborating not only with us, but also with each other and learn more about the other participants who are there. And we also have a special feature called the leaderboard. So if you earn enough points, right now you see that there are a lot of participants up here on the leaderboard who has been part of our previous programs and has earned a lot of points. And each of these participants have a completed profile. So you can actually relate to these different participants. And that is the utility of completing your profile. And going ahead, you can also add your social media links. There's an option to add your social media links. And for researchers, you can add your ORCID IDs, so that it's more easier for other people to find you. In case in the future, you would want to complete all the courses, share this link directly with somebody who would be interested in learning more about you and your achievements. So that is one of the reasons why you should have your profile complete. Now, once you have the profile completed and you're ready to begin, and we still have some time before the program starts on January 19, here are the coursework. So all participants who are registered will have a link to this page. And this is part of the organization page, and Ilya has already explained this. And we went through some of these courses here. So I saw that a lot of the participants from last time have started with introduction to bioinformatics, but I only saw that Godspar Obuli has been able to complete introduction to bioinformatics. And if you have already completed introduction to bioinformatics, you can start with the other free course, which is introduction to genomics. But just before the program begins, this would be good that most of us can complete introduction to bioinformatics and have an idea about what we are going to learn in this upcoming program. And this basic bioinformatics training is what we call level 1 program, where we introduce participants who are just starting to learn about bioinformatics to these different concepts, to multi-omics, and help them learn so that they can reach level 2. Yeah, so they can reach level 2. So what is level 2? When they do a more advanced program and design their own research project. So this is step 1, learning from the courses. Today, as I saw that there are a lot of questions from our participants, whether this coursework is going to be relevant to their research or not. So that is where the coursework is going to be very self-explanatory along with the sessions. And this will give you an overview, which in the future will help you develop your own project. So this is a very step-by-step process. That is why we start from the very basic introduction to bioinformatics, so that we can build up from step 1 to step 2 and above and beyond, right? And once you are done with the coursework through this program, we will introduce you to projects. So Ilya already mentioned the Ebola project, and there are other projects as well which are available. So once you finish all the coursework, that would be the next step, which is doing one of the projects which are here, or you can also propose your own project. Now, I would want to talk right now a little bit about the scholarship process. So we have received the applications for the scholarship, and I just wanted to let you all know that it is being processed. And the first step of the process is whenever you write down any of your references, we would want to set up a meeting with your references, introduce them to what we are doing here, and also learn more about you so that we can understand whether you are fit for the scholarship. Usually this scholarship is granted to undergraduate students, but in case there are any special circumstances, that is the reason for the meeting. So we are reaching out to you from Africa at pine.bio regarding all of your submissions. Please allow us some time. And if you're mentioning your references, you can also let them know that you're being part of a program, so that if we have time, if we set up a time for a meeting, you can then bring them, and we all can have a discussion together regarding your joining the program. And if you have any other questions, then please put them down in the chat box. And the next thing again that I wanted to share is what Adikunle, Aditayo, and Ilya has already been sharing, which is like doing your own project. So this starts in level 2, right? Once you finish this program and you are ready, you have learned about the different omics topics, you know about the logic, the application, you move on to proposing your own project, where you see there are different people who have already done their project. I have also shared this link right now in the chat box. You can go ahead and take a look, and all of these are individual presentation. So as Ilya was telling you, that you will not only be doing your project, but you will also be presenting the project where you will record a video like this, do a proper presentation, and we will be sharing the video on your behalf in our YouTube channel, which is watched by thousands of subscribers. We will share it in our groups, as you have already seen the groups that we have in LinkedIn, and here is our LinkedIn group, and also in Facebook. So once you are joining these groups and being part of the program, you can see that there is a lot of benefit. You are not only being part of just this program, but in the future, a part of a larger community who interact with each other, who share different research publications with each other. So you are able to learn and connect with an international community. And before I end today's meeting and presentation, I just wanted to let you know regarding those who had a question about the Bioinfo server. So access to this server is only created by us. So once you're registered for a program, you will receive an email which will have the details to log into Bioinfo server. And the username would usually be your email ID, but we will create the password for you. If you are not joining this program, but you still want to try out the demo pipelines, you can actually request for access. So once you request for access, you will have this form that you can fill up. And once you fill up the form, you will receive an email from my end, which is marketing@pine.bio, with the demo access to the platform and also some details about the different licenses that you can purchase to take to utilize the platform. So we do have educational licenses and research licenses. The research license gives you full access to the platform, which means that you are able to upload your own data files. And in case you are only looking to learn, we would definitely recommend you to join the program, because all of this is included within the program. The educational access, on the other hand, if you're purchasing it, is $75. So if you are looking forward to utilize this system and learn from the system, the best way is to join the program, start with the basic program, and then move on to the more advanced learning stage or the advanced project stage. And now, if you have any questions, this is open. So please let me know. Either myself, Ilya, or Aditayo and the African team can answer your questions.
Thank you, Bipsa. And I also wanted to mention, Aditayo, I put the link to the YouTube for you to have share with everyone in the chat. And I also will post the link to everyone so everyone can see the recording of this webinar. Great, thank you very much. Thank you everyone for joining. It was great to see all of you again, for those that are already registered. And we look forward to seeing you soon as the program starts. We also have some other resources that we will be sharing with you, and hopefully you will join our social media as well. So we'll stay in touch there and connect personally. Thank you.
Thank you, Ilya, and thank you everyone for joining again today. The trainings each day would be at 4:30 p.m. It's the same time, 4:30 p.m. WAT. So we have tried to make it in the afternoon so that it's easier for everyone to join. Thank you. And if you have any further questions, please reach out to africa@pine.bio for our team there to help you with any of your queries. And thank you everyone for joining again. It's a pleasure to meet everyone.